Close your eyes. Hi, Ram students. So we have completed with the two types immunity. Now I want you all to just revise the acquired immunity. So acquired immunity we were doing. Now I want Poonam to speak up. What is acquired immunity? Poonam,
Yes, ma'am.
what is acquired immunity?
Ma'am, it is a pathogen specific and it is characterized by memory.
Okay.
And ma'am, actually on that day when you taught this, I was absent.
So you were not there.
Yes,
Okay.
ma'am.
So who will
Sorry.
explain then?
Yes, ma'am.
Anjali. Anjali will tell. Anjali, are you there? Asta.
Yes, ma'am.
Can
Ma'am,
you
it is the
please?
immunity which
Huh?
we develop during the lifetime. It
Huh.
develops after exposed to antigens,
Okay, correct. Then what
and
else?
then its its characteristics are
Hmm.
diversity and specificity,
Correct.
and one important one is self and non-self discrimination. It
Correct.
can identify between self and non-self cells.
Okay.
And then one more characteristic is uh, that it has memory cells.
Okay, correct. Then what else?
They develop the memory of the antigenic response, which helps in generating a faster response to the antigen. It
Very good.
remembers the pathogen.
Sorry. Yes. Then next one.
They are pathogen specific,
Yeah,
which is known
pathogen
as
specific.
T lymphocytes and T lymphocytes.
Correct. Okay. Then.
They respond to specific pathogens by generating an immune response. That is, the first one is primary response, and
Yes.
if the uh, pathogen attacks again, then it remembers it and forms the memory cells, which attacks on the pathogen and saves our body from it.
Okay. Correct. So I'll be just presenting my screen, and we'll try to just see uh, whatever we have done, and then I'll take up the next topic, the antibody molecule. So first of all, try to see it. Now, active immunity and passive immunity, did you understand everyone? Active immunity and passive immunity. We did that also. Speak up. Can you all see my screen?
Yes, ma'am.
Okay. So active immunity and passive immunity, anyone can tell?
We haven't done it yet.
Acha, we have not done. Okay, so this was all which we have covered up. You have understood what are the two different types of responses. You all have noted the differences also. Yes or no?
Yes, ma'am.
Okay.
Yes, ma'am.
Then what are the two types of the lymphocytes? Can anyone tell?
Ma'am,
T
B lymphocytes and
lymphocyte
T
and B
lymphocytes.
lymphocyte.
Okay. Why you call them as T lymphocytes and B
Ma'am,
lymphocytes?
because they get matured in the thymus.
Thymus, correct. So you call them T lymphocytes. And why we call them B lymphocytes to some
Ma'am, because they get matured in the bone marrow.
bone marrow itself. Okay. What is the difference in their action mechanism?
Ma'am, T lymphocytes attack the pathogen and release cytotoxic chemicals to kill it.
Sorry.
And B lymphocytes produce pathogenic specific immunoglobulins called antibodies.
Antibodies. Okay. So I hope this is very clear to everyone. So this was all which we have done. So the natural immunity and the artificial immunity you understood?
Yes, ma'am.
So artificial we did, they are of two types. So quickly tell me, this last time also I have discussed. Artificial immunity we find of two types. What are the two types?
Ma'am, only get one vaccination.
Active immunity. So I was telling you, just note down, children. It's very, very important that what is the difference between active immunity and passive immunity. Active immunity means that when your immune system gets activated by injecting the partly active or attenuated pathogen or the parts into an individual to generate an immune response. So you call that as active immunity. Is what is happening, your immune system is getting activated and it is producing the antibodies and the memory cells. Followed to you call it as active immunity. And what is passive immunity? That is, you find that the immune system of the body will not play any role. From outside, the prepared antibodies are given into the body, injected into the body. Did you get it? For example, whatever we are taking the vaccines, BCG, and then your polio vaccine, DPT, they will be inducing or they will be developing active immunity. Okay, but suppose if someone makes an accident and then means that some snake bites, so what will happen? To neutralize the poison of the snake, the doctors will give the that antitoxin. Okay, and that will be means neutralizing that poison. तो उसमें क्या दिया जाएगा? Prepared
Ma'am, your voice
antibody.
is not clear.
Voice is not clear. Let me
Yes, ma'am.
check. My mic is. I'll make it full. Can you hear now?
Yes, ma'am.
Okay. Better now?
Yes, ma'am.
हाँ, सो एक्टिव इम्यूनिटी अच्छे से समझ लो। एक्टिव इम्यूनिटी में क्या कर रहे हैं हम? इम्यून सिस्टम को एक्टिवेट कर रहे हैं, इम्यून सिस्टम ऑफ द बॉडी। ठीक है, और कैसे एक्टिवेट कर रहे हैं? बाय गिविंग इंजेक्टिंग द एटीनुएटेड पैथोजेंस और देयर पार्ट्स। तो व्हाट विल हैपन, जैसे ही वो पैथोजन पार्टली एक्टिव पैथोजन बॉडी में पहुंचेगा तो बॉडी का इम्यून सिस्टम एक्टिव होगा एंड विल प्रोड्यूस द एंटीबॉडीज एंड विल डिस्ट्रॉय इट। ठीक है, लेकिन पैसिव इम्यूनिटी में क्या करते हैं हम? हमारे बॉडी के इम्यून सिस्टम का कोई रोल नहीं रहता, हम प्रिपेयर्ड रेडीमेड एंटीबॉडीज को बॉडी में डालते हैं जो उस पैथोजन से लड़ता है। ओके, तो स्नेक बाइट में जो एंटी वेनम दिया जाता है दैट विल कम अंडर द पैसिव इम्यूनिटी, इट विल डेवलप पैसिव इम्यूनिटी। डिड यू गेट इट? तो पुट टुडेज डेट एंड राइट डाउन artificial immunity and put a dash and write down types.
Ma'am,
Yes, sir.
ma'am, if there is same treatment for any snake bite or different treatment for different snakes?
It may be different, but generally you find that doctors have only one anti venom. Okay, up specifically, so I don't think that they have, because they will not. No one understands, no one identifies that which snake has actually bitten. Na? That is also
Yes,
one thing. हाँ, सारे लोग पहचानते क्यों है कि कौन सा स्नेक ने काटा है? वो तो बस ये है कि दे हैव अ स्टैंडर्ड एंटी वेनम एंड दे इंजेक्ट इट एंड दैट वर्क्स फॉर इट। ओके, Uh-huh.
Okay.
So what are the two types of immunity? Did you understand or not?
Yes, ma'am.
Huh. So
Yes,
I,
sir.
I want someone to explain in the active immunity what is done and what is passive immunity. And then you have to make the differences. You have to write down the differences between the two. It's very, very important for the exam. Ajay, tell me one thing. You all have got one refresher everyone?
Yes, ma'am.
Everyone has.
No, no, ma'am.
No, ma'am.
Who is not having?
Ma'am, this
Ma'am,
year we don't get.
Nandini.
Huh? Again. Because
Ma'am, Nandini.
Nandini only. Rest everyone has got.
Ma'am, me
No,
also Poonam.
No, ma'am.
Ma'am,
ma'am.
फिर किसी को भी नहीं मिला था क्या?
नहीं मैम, इस साल नहीं मिला, टाइम ही कम मिला, 2 दिन तो स्कूल ही आए थे।
हाँ वही दैट ओनली आई एम आस्किंग। तो 2 days में अपना वो काम नहीं हो पाया था ना?
हाँ मैम।
अरे तो फिर मतलब आप लोग के लिए पढ़ने की दिक्कत होगी, isn't it? If I say also that you go through the means you have the textbook. If you would have taken one refresher, it would have been very nice. So you would have studied everything nicely. Chalo, so then whatever we are doing in the class na, that only you'll have to note down. Okay. And when we will be meeting, so then I will be giving you one refresher. Okay. Not speaking.
ओके मैम।
Okay. So
okay.
active immunity, passive immunity, everyone note down. Okay. So artificial immunity, how many types? Two types. What are those two types?
एक्टिव एंड पैसिव।
Active and passive. What is active immunity?
Pathogens are injected
Huh?
so that body will generate its immune response to fight the disease
Huh.
later on.
Correct. Okay, it will fight it that time also and it will produce the memory cells also. And that will help in further preventing the same disease. So this is the basis of the vaccination. Okay, and what is passive immunity?
Ma'am, prepared antibodies are
Huh?
injected directly so that
Huh?
it will work directly, and
Huh?
no immune. There was no work for
Huh? Immune system of the body will not play any role in it. Okay, so which one will be faster you think?
ma'am, passive immunity.
हाँ, पैसिव इम्यूनिटी विल बी फास्टर। एक्टिव इम्यूनिटी के लिए तो वेट करना पड़ेगा जब इम्यून सिस्टम प्रोड्यूस करेगा एंटीबॉडीज, बट दिस इज वेरी फास्ट। ओके, सो वी हैव दैट। टिटनेस के लिए भी एक एग्जांपल नोट कर लेना, दैट आर्टिफिशियली प्रोड्यूस टिटनेस एंटीटॉक्सिन इज इंजेक्टेड टू प्रिवेंट टिटनेस, एंड वन आई गिव विद एग्जांपल स्नेक बाइट। Okay, so now active immunity and vaccination. What is the principle behind it? That you all know, so shall we skip this thing? Okay, vaccine generation they have given. This you can note it for just your general knowledge. The questions will not be asked in the board exam. Okay, the vaccines, they are first generation vaccines, second generation vaccines, and third generation vaccines. Okay, so they are produced by conventional methods from inactivated pathogens. Means if inactivated pathogens are injected into the body or weakened pathogens, तो हम उसको बोलेंगे फर्स्ट जनरेशन। ओके, अगर वो रिकॉम्बिनेंट डीएनए टेक्निक से वैक्सीन तैयार होती है तो वी कॉल इट एज सेकंड जनरेशन वैक्सीन। एंड इफ the vaccine contains na synthetic peptides, carbohydrates or antigens, so you will call them, and those antigens or synthetic peptides किसका होगा? They will be taken from the bacteria or the virus. Okay,
Ma'am,
the
we are
culture.
using all three.
Huh?
Ma'am, we are
Correct?
using which generation vaccine?
We are using all the different types for different diseases. Okay, so you'll have to further means get the information that polio drop कैसा है, ओके, फिर बीसीजी कैसा है। मोस्टली द पार्टली एक्टिव वंस आर यूज्ड। फिर अभी जो वैक्सीन बन रहे हैं वो कल्चर और बैक्टीरियल और वायरल कल्चर से प्रिपेयर कर रहे हैं बाय एक्सट्रैक्टिंग देयर सिंथेटिक पेप्टाइड्स आल्सो। So they are considered safer. Okay.
Okay.
So shall I take up the next one? So passive immunity, the
Ma'am,
examples are given. Huh?
I have a doubt.
Say.
Why some vaccines are given by a drop like polio and some are injected?
Some are injected. So that now see if you are injecting, so what will happen? It will go into the blood, isn't it? अगर ओरल ले रहे हैं तो फिर वो अपने डाइजेस्टिव ट्रैक्ट में जाएगा, तो देन डाइजेस्टिव ट्रैक्ट से वो फिर सिस्टम तक पहुंच पाएगा. Now they must be producing a actual reason. We'll have to see that they want it to directly go into the blood, so they will inject. And if they want some pathogen to go into the that your uh, digestive tract, they will be taken orally, like the polio drop. Okay, but ultimately they will be inducing the immune system. Now you can note down this thing. Passive immunity involves inserting pre-formed antibodies into the human body. Write down this point. Passive immunity involves. Did you note down the previous one?
Yes, ma'am.
Okay. So passive immunity, add up this point also. It involves inserting pre-formed antibodies into the human body. Okay, so it may be
Ma'am, these
from mother.
antibodies are also then taken up from the human.
It can be taken up from the humans. Why not? From one human and it will be given to the other person. Okay, that can be done. For example, for this corona, they are doing the plasma therapy.
Plasma therapy.
Plasma therapy is this only. It will be passive immunity. ओके, इट विल डेवलप द पैसिव एंटी, क्योंकि वो प्लाज्मा में एंटीबॉडीज पहले से रहेंगे और वो जाके सीधे सीधे उस वायरस से फाइट करेंगे. So that will come under the passive immunity. Okay, so they have given the diseases also. It is used against diseases such as measles, mumps, rubella, tetanus, scarlet fever, and rabies. Okay, and also against the snake venom. You can note down the examples where we are using the passive immunity, where we are developing the passive immunity to cure the disease. Noted?
And wait,
Huh. As you note down, just tell me that next, ma'am, so I will be able to change it. Examples you note down.
Ma'am,
Yes.
ma'am, what happened in mumps?
Mumps, you all know your salivary glands get affected. Okay, and the tonsils. Mumps, actually, remember we have the tonsils in the neck region.
Yes,
Okay,
ma'am.
and tonsils we'll study in detail. These tonsils are the site of fight between the pathogen and the immune system, that is the antibodies produced by it.
Yes,
ओके, तो सबसे पहले ना वहाँ पर टॉन्सिल्स में भी फिल्टर हो रहा है, कोई भी पैथोजन डिस्ट्रॉय किया जाता है. तो when the pathogen is little stronger and the fight is going on, so these tonsils they swell up. Okay, and that swelling and inflammation of the tonsils only we call it as the mumps. तो देखा होगा
ma'am,
कुछ लोगों को गले गले में वो एक पट्टी जैसी चिपकाते हैं ना, कुछ स्टिकर जैसा,
yes,
मालूम
ma'am.
है? वो
Yes, ma'am.
Yes, ma'am.
छोटे छोटे उसमें होल्स रहते हैं, वो लगा देते हैं.
Yes,
Yes,
ma'am.
ma'am.
वो मम्स के लिए ही रहता है, तो वो टॉन्सिल्स का इन्फेक्शन है. मम्स इज अ इन्फेक्शन ऑफ द टॉन्सिल्स. ओके, और इन्फेक्शन एक्चुअली वो होता क्यों है? क्योंकि वहां पर वो फाइट चलती रहती है, और जब वो फाइट जीत नहीं पाता है या कुछ लंबा समय चलता है, तो फिर देन दे गेट inflamed. Okay, so now shall I take up the next one?
yes, ma'am.
Yes, ma'am.
Okay. So differences you can note down between the active immunity and passive, developed by the body in response to an infection or a vaccine, and developed when pre-formed antibodies are inoculated into the body. Okay, so that way source you can write down, then side effects. Active immunity has got no side effects. ओके, बिकॉज इम्यून सिस्टम, हमारे बॉडी का इम्यून सिस्टम उन एंटीबॉडीज को बना रहा है, so that's why they will have no side effects. Okay, but in the passive immunity we are injecting the prepared antibodies, that is the pre-formed antibodies. They may cause some reaction. Noting everyone?
Yes, ma'am.
Okay. So all the 4 points of difference write down. It's very, very important. And active immunity, it is slow, provides relief after a long period, and the passive immunity provides immediate relief. Provides immediate relief. But वो relief ना short time के लिए होगा. So this is short lived. But your active immunity, because your immune system has got activated, and it will form the memory cells also, so it will be long lasting. Which one will be long lasting?
Active immunity.
Active immunity. Okay. Yuvesh, noting down.
Yes, ma'am.
Everything you have to note down. This will help you in learning the things then later. You are reading the textbook also,
Yes, ma'am.
everyone. And if anything na in the textbook you don't understand, better ask. Okay. पर यहाँ पर समझने के बाद आपको बुक में भी पूरा समझ में आ ही जाएगा. Shall I take up now next?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. So now I end up this thing. We will move back to the new topic. So you know that it is means your immunity is due to the uh, immune system is working. Uh, the whole mechanism is dependent upon the antibodies. Okay, so now we will take up the antibody molecule, its structure. Did you all see in the book, children? One Y-shaped antibody cartoon is made. Okay, Y-shaped structure is there.
Yes, ma'am.
So that is the antibody molecule. Everyone will draw it. Okay, it is having the heavy chain and the light chain. In the book, the diagram is showing very nicely the Y-shaped structure. Not opening, what happened? Okay. So first we see this, then I'll show you. So H1, did you get that? Did you all read in that book?
Yes,
So
ma'am.
B lymphocytes and T lymphocytes. Let you try to see this. Okay, this all we have done, the active and passive one, pathogen specific. Lymphocytes, this we have discussed already. So this point you mark it, the T lymphocytes, they form about 75% of the circulating lymphocytes. Write it down about the T lymphocytes. that they form about 75% of the circulating lymphocytes, and they are formed in the thymus, okay, not in the bone marrow. Okay, differentiate now. Next, owing to the presence of hormone called thymosin, they undergo differentiation in the thymus and become immunological competent, but they are formed in the bone marrow. That way it is correct that formation तो वहीं होगा, but differentiation कहाँ होगा? Where the differentiation and maturation will take
Thymus.
place?
Thymus.
Thymus. Differentiation will take place in the thymus. Now, what do? So they help the B cells in producing antibodies. Noting everyone?
No, ma'am.
Note down. And then what do? They generate a cell mediated immune response. They generate a cell mediated immune response.
Ma'am, what is B cells? What are B cells?
B cells are those which are formed in the bone marrow itself. वो बोन मैरो में ही मैच्योर भी होते हैं, डिफरेंशिएट भी होते हैं और बनते भी हैं. ठीक है, लेकिन टी सेल्स बनते तो बोन मैरो में है, पर उनका डिफरेंशिएशन मैच्योरेशन कहाँ होता है? क्या होता है उनका डिफरेंशिएशन और मैच्योरेशन?
Thymus.
थाइमस में. मतलब उसके लिए उनको वहां जाना पड़ता है. तो मीन्स थाइमोसिन के प्रेजेंस में इनका डिफरेंशिएशन होता है. So that's why, because thymus is playing an important role in the differentiation, you are calling them T lymphocytes. Okay, and they help the B cells in producing antibodies. And what type of the immune response we call it? What type of the immune response do they generate?
Cell-mediated immunity.
Cell mediated immune response. Okay, noted everyone? Okay, shall I take up the next one?
Oh, yes.
हाँ, जल्दी जल्दी करते जाओ, शॉर्ट में लिख लिया करो. Okay, and B lymphocytes now, they differentiate in the lymphoid tissue of the tonsils, pairs, patches, and appendix, and they are independent of the thymus. So note down this thing. B lymphocytes, they differentiate in the lymphoid tissue of the tonsils, pairs, patches, and appendix, and are independent of the thymus. तो रिमेम्बर, बी लिम्फोसाइट्स, वो लिम्फोसाइट्स बोन मैरो में बन के कहाँ पहुंचने वाले हैं? इन जगह पर टॉन्सिल्स में पहुंचेंगे, पेयर्स, पैचेस एंड अपेंडिक्स, एंड they will not be affected by the thymus. They will have no role in the differentiation process of the thymus. Is it very clear this point? Speak up, students.
Yes, ma'am.
Yes, ma'am.
Okay. Now what they do? They respond to pathogens or antigens. They release proteins that are generally called antibodies. So they produce the antibodies. The antibodies are the type of proteins only. Okay, then antibodies. What are the antibodies? So they are the special proteins with 4 peptide chains, 2 light chains, and 2 heavy chains. So that's why now we denoted by writing denoted as H2L2. How is the antibody molecule denoted? H2L2. How is it denoted?
H2L2.
Yeah, why H2? Because 2 heavy chains. H is standing for heavy and L is for the light chain. तो light chain कितने हैं? 2 हैं. Now, what are the different types of the antibodies? Note it down. IgM, IgG, IgA, IgD, and IgE. So, how many types? Astha, how many types of antibodies?
Five.
Five types of antibodies. And how do we denote the antibody molecule?
And they are
How do we
denoted
denote?
as X2 and X2 and 2.
Make your mic proper. Okay. And what type of the immunity do they generate? Humoral immune response. So humoral immune response. And the previous one, T lymphocytes are producing which type of the immunity or immune
Cell
response?
mediated.
Cell mediated. This should be very clear to you. So sometimes
What's
in
the
the
mean of
exam they
humoral?
हाँ I'll tell you. Now see, सेल मीडिएटेड मतलब कि सेल्स ही पैथोजन से लड़ रहे हैं तो सेल मीडिएटेड हो गया. ओके, अब इधर पर क्यों? क्योंकि वो बी सेल्स को हेल्प कर रहा था एंटीबॉडीज प्रोड्यूस करने में, तो बी सेल्स वर डूइंग द वर्क देयर. यहाँ पर क्या हो रहा है? बी सेल्स जो है वो क्या कर रहे हैं? एंटीबॉडीज प्रोड्यूस कर रहे हैं. एंड एंटीबॉडीज आर द टाइप ऑफ प्रोटीन्स विच विल कम इनटू द प्लाज्मा. एंटीबॉडीज कहाँ पहुंच जाएंगे?
Plasma.
प्लाज्मा में. एंड प्लाज्मा में पहुंचते हैं बिकॉज प्लाज्मा इज अ लिक्विड, वी कॉल इट एज अ humoral immune response. So immune response is shown by the plasma. That's why this type of the response we call it as humoral immune response. Or previous one is known as cell mediated immune response because it is shown by the lymphocytes itself. Then
Yes, ma'am.
so differences you will be able to write if it is asked in the exam.
Yes,
Everyone,
Yes, ma'am.
keep up children. Right. If the question is asked by the difference between T lymphocytes and B lymphocytes, you got the differences
yes, ma'am.
and all the differences are clear to you.
Yes, ma'am.
Yes or no? जल्दी बोलते
Yes,
जाओ तो
ma'am.
फिर आगे बढ़े हम लोग. Okay. Shall we take up the next?
Yes, ma'am.
Okay. अभी हम लोग को स्ट्रक्चर भी पढ़ना है ना. So humor B lymphocytes, they have given again the same thing. This part you write it out. That is, through the humoral immune system, the antibodies fight with the antigens by circulating in the lymph. Okay, the lifespan of B lymphocytes is very short, and soon after the death, they are replaced by new cells. Okay, तो lifespan short है, ये बस note कर लो. Okay, and antibodies are produced by the plasma cells at the rate of 2000 molecules per second. सोचो तो कितना fast produce होते हैं. Okay, this can be also noted that B lymphocytes come in contact with an antigen, they divide rapidly to form plasma cells. जो multiply होकर lymphocytes बनेंगे उनको हम क्या बोलेंगे? What you'll call them? Plasma cells, children. What you call them?
Plasma cells.
Plasma cells.
Plasma cells. हाँ. Are you understanding what we are discussing? एक चीज क्या याद रखती है कि बी lymphocytes, they will, when they'll come in contact with antigen, so they will divide, that is multiply rapidly to form plasma cells, and then plasma cells will produce antibodies. Plasma cells will produce
Antibodies.
antibodies at what rate?
2000
200
molecule per
molecules per
second.
second. 2000 molecules per
Okay.
second.
हाँ, and वो antibodies कहाँ पहुंच जाएंगे?
Plasma.
Antibodies will go into the lymph and will go into the blood plasma. That will go into the lymph and the blood plasma. Is this much very clear to you all?
Yes, ma'am.
No, noted children?
No, ma'am.
हाँ, so note it down. शॉर्ट में नोट करो, सिर्फ पूरा पूरा मत लिखना, सिर्फ इतना लिखो कि बी lymphocytes, they divide rapidly when they come in contact with antigen and form plasma cells which produce antibodies, which produce antibodies. Antibodies which go into the lymph and the plasma and fight with the antigens. And fight with the antigens. Alima, are you noting the things?
Yes, ma'am.
Okay. Understanding also?
Yes, ma'am.
Okay.
Done.
This is important. This is important topic for the board exam, you know. Done?
Yes, ma'am.
Shall I take up the next one?
Yes, ma'am.
Okay. Now, the T cells are stimulated. So this you write down. They are of different types. So T cells, what are natural killer T cells, helper T cells, and suppressor T cells. Okay. T lymphocytes are of three different types. They are divided into three different classes based on their function. So, natural killer T cells, and short you write down that they release lymphokines. They release lymphokines. किलर सेल्स कहाँ जाएंगे? जहाँ पर कोई इन्फेक्शन है वहाँ पहुँच जाएंगे. दे माइग्रेट टू द साइट ऑफ infection and release the lymphokines. And helper T cells, second class is what? Helper T cells. तो नाम ही जुड़ा हुआ है helper, मतलब they will be helping the B cells. They will help the B cells to produce more antibodies. They help the B cells to produce more antibodies and enhance the activity of the killer T cells. Enhance the activity of killer T cells.
Ma'am, what is
Noted?
lymphokine?
Lymphokine is a chemical. Lymphokine is a chemical which will be produced और वो इन्फेक्शन को खत्म करेगा. वहां पर प्रेजेंट साइट ऑफ इन्फेक्शन, उस जगह पर जो भी पैथोजन प्रेजेंट होंगे उनको डिस्ट्रॉय करेगा ये केमिकल. ओके. You know that T cells तो cytotoxic substance produce करते हैं, that is only lymphokine. Okay, and helper T cells are helping the B cells to produce more antibodies and enhance the activity of killer T cells. And suppressor T cells, what they'll do? They secrete suppressive factors. Secrete suppressive factors which suppress the immune system and protect the body cells from being attacked. मतलब इम्यून सिस्टम क्या हो सकते हैं? कुछ एंटीबॉडीज जो बॉडी की है वो क्या करेंगे? कई बार बॉडी के ही ओन सेल्स को भी डिस्ट्रॉय कर सकते हैं. दे कैन अटैक द बॉडी ओन सेल्स. So the suppressor T cells will not allow the immune system to attack the own cells. That is, so protecting, this way they are protecting the body cells from being attacked. So this is known as immune tolerance. Immune tolerance. कई बार ऐसा होता है चिल्ड्रन, दैट वी कॉल इट एज ऑटो इम्यूनिटी. बॉडी स्टार्ट द इम्यून सिस्टम स्टार्ट्स अटैकिंग द ओन सेल्स। इसमें तो समझ जाओ कितनी दिक्कत हो जाएगी अगर बॉडी का इम्यून सिस्टम अपने ही सेल्स को डिस्ट्रॉय करने लग जाए।
मैम
इट इज वेरी
एलर्जी में भी
डिफिकल्ट।
एलर्जी में होता है ना ऐसा? तो
एनर्जी में कैसे?
एलर्जी, किसी चीज से एलर्जी होती है तो इम्यून
एलर्जी,
रिस्पॉन्स
हाँ,
करता है ना इम्यून
एलर्जी
सिस्टम।
इज नो, एलर्जी इज हाइपर सेंसिटिविटी। Okay, allergy is hypersensitivity. That is a different thing. Hypersensitivity of the immune system. कि जो चीज वैसे तो नुकसान की नहीं है, बट क्योंकि वो इम्यून सिस्टम उसको एक्सेप्ट नहीं करा, उसको उसके लिए हाइपर सेंसिटिविटी दिखा रहा है। ओके, सो इट स्टार्ट्स प्रोड्यूसिंग द डिफरेंट सिम्टम्स देन। ओके, सो एलर्जी इज नॉट द इन दैट द इम्यून सिस्टम इज नॉट अटैकिंग ओन सेल्स, ऐसा नहीं है। ओके, so suppressor T cells, did you understand? Noted?
यस मैम।
Okay, so I'll take up the next one. The differences between the B cells and T cells. So already you have noted that. Okay, so we'll take up the, all points are clear to you? B cells and T cells means ये वाला point clear हो रहा है, humoral, cell mediated, you have already noted. Then where it is differentiating, you have noted. Acts against bacteria, viruses, and it acts against pathogenic microorganisms, transplants, and cancerous cells also, and produces clones of plasma cells. It produces antibodies, produces clones of T cells. And they do not suppress the immune system, but they suppress the immune system. Which one suppress the immune system?
बी lymphocyte।
B lymphocyte.
The points which you have not covered, that point you can write down. This point you can add up. The other points that we have already done. Okay, do not suppress the immune system, but these suppressor cells suppress the immune system. Done?
Done.
Okay, we have added this difference in the previous table which you have written between the B and T. Everyone?
Yes, ma'am. Ma'am.
Okay. So now we'll start with the, so put the subheading in your notebook, structure of the antibody. Structure of an antibody. So the antibodies, you write down that they are produced by the B cells in response to an antigen. It is produced by the B cells in response to an antigen. And it is Y-shaped. It is Y-shaped tetrapeptide protein. It is Y-shaped Y-shaped tetrapeptide protein is made up of 4 chains, okay, peptide chains, formed of 2 identical light chains. So which are the light chains? Can you make it out here in the figure? Can you see my cursor? Yes or no? हाँ, तो ये जो पूरा ये कंप्लीट वाई का शेप दे रहा है, दैट इज योर हैवी चेन। ओके, और जो सिर्फ एक छोटा सा पार्ट जो आपके अपर पार्ट से वाई के अपर पार्ट में जॉइन हो रहा है, दैट इज योर लाइट चेन। ओके, सो 2 heavy chains, दैट्स व्हाई 2 identical लाइट चेन्स, दोनों सेम है, और 2 सेम हैवी चेन्स है। Okay, now these 2 terms are not given in your book, but you can note down the 2 parts they are calling paratope and epitope. Which is the paratope? The hypervariable region is a part on the antibody which binds the antigen. So ये वाले part को what you are calling paratope. Okay, and this is the part, the top part which is showing this yellow part. This is the antigen binding site. Means any pathogen, antibody जब पैथोजन से कांटेक्ट में आएगा तो इस जगह से वो बाइंड हो जाएगा। Is it clear? Okay, then epi2, what is this? The corresponding region of the antigen that is in contact with the palato. तो ये बाकी का पार्ट जो है आपका, that we call it as the epitope. So two light chains along with two heavy chains of the antibody, they are interlinked. तो यहाँ दोनों के बीच में कनेक्शन क्या है? They are joined, they are interlinked through the disulfide bonds. They are connected or interlinked through the disulfide bonds. So therefore, there are how many disulfide bonds? There are 3 disulfide bonds in an antibody. Did you understand, Tika?
Yes, ma'am.
Okay. So the same structure is given in your textbook also. Everyone will draw that antibody molecule in the notebook. Okay? And very, very important for your board exam. They ask in the just to draw the antibody molecule. Shall I take up the next one?
Yes, ma'am.
Okay. So recap so done. I'll take up to the next now. So session is timed out. So already 49 it is. So I'll take up once again. I'll stop sharing now. And now next period I'll take up once again the antibody structure, and everyone will do one work that you will draw the diagram of the antibody molecule in the notebook and revise this whole thing.
Yes, ma'am.
Okay? And then we'll start with the lymphoid organs. So
Yes,
try
ma'am.
to read the lymphoid organs, students. The next period will take up the lymphoid organs. Okay. So now we'll do Shantipat. I'll just take up once again your attendance. Then I'll stop the recording.